Hello and welcome to the AnoExpress tutorial. So AnoExpress is a Python package and a meta-analysis of RNA sequencing studies that have investigated insecticide resistance in Anopheles gambiae, sentillato, and Anopheles funestus. And in this video, I'm just going to show you, give a walkthrough of some of the functionality in AnoExpress, which may help you to use the tool itself. So we'll begin with, basically, you can access the AnoExpress notebooks from the GitHub repository. There are various badges here which link out to Google Colab. If you're unfamiliar, Google Colab is a platform that Google runs, which allows individuals to use computing resources in the form of Jupyter-like style notebooks for free. And so the first one I'm going to show you is the plot gene expression notebook, which is essentially has functions in it to plot gene expression, to plot fold change data and count data from genes of interest. And so the first thing we need to do is just install AnoExpress onto the Google Colab environment, and we will then import the package. So it's worth noting that you don't need to use AnoExpress in Google Colab. As with any other package, it can be installed with pip install on any local computer or compute cluster. But I'm going to show you how to use it in Google Colab for now. And so that is now installed, and we now have imported the package. So to begin with, we'll use this form essentially in AnoExpress, because we are essentially trying to, on occasion, compare between different species. We needed to find orthologs between the different reference genomes for Anopheles gambiae, Anopheles colutzii, Anopheles arabiensis, and Anopheles funestus. And everything is based on the Anopheles gambiae pest reference genome. And so there are various different analyses for that. So you have just the GAM-COLU analysis, which contains only experiments of Anopheles gambiae and colutzii, a further one with arabiensis, and a further one with funestus as well, and then one with funestus just on its own. The reason for that is, as we incorporate more and more data from more disparate species, we actually lose the amount of genes in our data set because we can only include one-to-one orthologs. So let's begin with GAM-COLU Arab fun analysis, and we're going to run this, which I believe is CYP6P3, which is a gene that is known to be involved in pyrethroid resistance in Anopheles gambiae. And in this case, for funestus, the values will be derived from CYP6P9A and B, which is the ortholog of CYP6P3 in Anopheles funestus. We can do this kind of strip plot. So we have log2 fold change data on the left. Each one of these dots is a single experiment. And if we hover over the points, we get information on that experiment. What was the resistant strain? What was the susceptible strain? Which country did it come from? And what was the exact log2 fold change? And also we have then the count data. So this is from the RNA sequencing reads themselves. How many read counts were assigned to this gene? And this is useful because some genes are very lowly expressed by nature, some are sort of more highly expressed, and so this is one way of evaluating that. We can also include microarray data from previously published studies by ticking this box, and if we rerun that now, there'll be less data points here. And we can also produce box plots as well, just like that. So now to show you some of the other features you can do, we can provide lists of gene IDs to produce a plot for multiple genes. We can also filter on a given p-value, so this will only plot points if the p-value in that experiment was less than 0.05. There we go, and it's pretty quick, the functions in general. So these are 2 carboxylesterases which I've recently published on. So another thing that we can do is we can plot, give the function a region instead of a gene ID, and it's going to find all the genes that are located in that region and plot gene expression for those. So let's give that a go. And in the meantime, I'm just going to make a new file which has a list of gene IDs. So let's take these genes. And let's save that onto our desktop and call it my gene IDs dot xaccess. Okay, saved, and let's quit that, quit that, quit that. Okay, so we can see that this data has been plotted now, and these genes within the CYP6P region have been plotted in their by their position. So we have CYP6AA1 and 2, carboxylesterase, and then going onwards. There is other functionality in this function. For that, I would just recommend having a quick look in the documentation. So if we look at the API, there is various things you can do. So as I mentioned, you can filter on p-values. You can sort by the name of the genes, or you can sort by the mean or median values of the fold change data. And you can also add titles and save as a HTML the interactive plots. So let's try now and upload that file which includes gene IDs. So say we have specific genes we're interested in, and we want to make a plot with those. First of all, we need to upload that file into Google Colab. For that, you go to this tab, Files, click on this little icon, Upload, and now we have that gene here, that Excel file here. So let's take this and and so what we do now is essentially we just pass the path of that file, and it can be an Excel file. It should also be able to be a TSV, tab separated file, or a CSV, and they should all work. And so if it's been a while since I did this, but if I'm correct, testing file input, if I'm correct, this will work. It does not work because I have missed the brackets out. Taking a little longer, I think, because we're sorting by position potentially. We have to load the GFF data itself, and so to give it 2 more seconds. Really shouldn't be that long. To be honest, it should just take a couple seconds. And there we go. So we have our list. We've provided our list of genes. We don't want in that file. We want a single column. The first column should be AGAP gene IDs. No headers in that file to be expected. Literally just the genes themselves, nothing else. And we plot them. It's going to plot them the minute it's sorted them by position now potentially, and we can see like there's some genes here which have really high read counts. A couple of them. It's like 9K1 is one of them. So awesome. So I have showed you how to use the functionalities within Plot Gene Expression Notebook. Let's go on to something else and have a quick look at some of the other things we can do. If I go on the utility functions, all of the data within AnoExpress is there for you to use, for you to load, for you to play with, for you to do further analyses on. So let's give that a go. So I'm going to open a new Colab notebook. We could have done it in the same one, but let's use this one. And let's take a look. So there is essentially, if you want to load the data from AnoExpress, there's one main function, which is data, which we can use. And so we can load the fold change data here, we can load the p-value data, and we can also load the read count data itself, specifically the log2 count data. And so we'll just let AnoExpress install again. Again, if we can load the microarray data if we're looking for fold change data or p-values. Okay, so that's done. So now let's do this function. We're gonna, for this analysis, the Gamma-Colu Arab fun analysis, we want to load the fold change data, and let's have a look what it looks like. So there we go. This is just the first 5 rows of that data set, but if we take a look, we can see that there's 8,599 genes in total and 28 experiments. You know, if we load the microarray data, then we will see that there is now 59 experiments. And hopefully we'll add to these as well. And if you're interested in adding a data set to AnoExpress, a more recent data set, please just make an issue on the GitHub repo and we will look to incorporate that. As I said, you can do the same for p-values. You can do the same with microarray data. You can also use the function metadata to load the comparison metadata. So there's 2 different types of metadata in AnoExpress. There's the comparison metadata. So this is sort of one row for each differential expression contrast. So you have the comparison TF4 versus Nguso. Tells you what the resistant strain was, what the susceptible strain was, what species it is, what country, and what latitude, longitude. And you can also then load the sample metadata. So let's give that a go. Express sample metadata and the analysis. I think let's do Gamma-Colu, and that will then load 1 row for every single sample. So we can see that for that analysis there's 152 samples. For if you include the phonesis, there's now 198 samples. And so this is just handy to help load the data, these functions, and inspect the metadata as well. Now, there are various other things that you can do with AnoExpress. You can make gene regulatory networks. You can plot heat maps of the data. You can also perform gene set enrichment analysis. Something that you may want to do, and I'm happy to talk about if people have specific questions, please let me know and I can talk to you about using AnoExpress. You can also reproduce the actual differential expression analysis that was done for the meta-analysis in the first place. So this is in R with DESeq2, but you can run R as well as Python, Google Colab. And so it's quite cool to be able to reproduce that whole thing. All of the input files are stored in the GitHub repository, and so you can recreate that. Losing my voice, it seems. In this notebook, I'm not going to run it because it's already run, but there are functions called load candidates, which will essentially, for each, give it an analysis and give it a function to summarize the data with. So in this case, we're going to calculate the median. it will summarize for each gene what the median or mean log2 fold change is, and that way you can then rank genes, trying to find resistance candidates, which is something that we did for the AnoExpress manuscript. This function looks at consistently overexpressed or underexpressed genes. So, for example, in this function, we're trying to find genes which are downregulated in 20 out of all experiments. Similarly, you might want to do the same, but commonly overexpressed ones. So here we're looking at genes that are expressed in 24 out of 31 experiments. And you can see there's some candidates that we know. SLIPE6P3, SLIPE6Z2, GSTDD3, SLIPE9K1, and so on. Something SLIPE4H17 is something that came up a lot in the paper. So that's another example of something that you can do. And also here, we also can perform gene set enrichment analysis on the data, either with Go terms, with PFAM domains, or also KEGG pathways as well. So I hope that's useful, and if you've got any more questions, please just email me, or my email is just here, or raise an issue on the GitHub, and I'll be happy to help or discuss further. Thank you very much for listening. Have a good day.